but I get sometimes if I talk fast or you miss something or you get distracted by whatever else is going on in your home right now, that it might be nice to review the recording. And so, yeah, we can do that. But it's not to be used in lieu of attending, okay? Yeah. All right. So we're recording now, and there's the chat. Okay, yes. And then, so let me share my screen again. Where did it go? There we go. Try to be on time. We only have 50 minutes on Monday and Wednesday, and then an hour and 25 minutes on Friday for lecture. And I'm used to lecturing for 2 hours, so I don't like crunching it down into 50 minutes because the time just flies by. And so, you know, I don't want to waste a lot of time. I want to be able to jump into the lectures as quickly as possible. So try to be on time and try to be prepared. So my kindergartner and my third grader, the teachers make them where they say, you know, your kids should be dressed and fed before class time, right? And, you know, I think that that's up to you. Not everybody eats breakfast. I'm not going to require you to have been, to feed yourself, but, you know, if you want to be in your pajamas, that's fine. If you don't want to wear pants because all we see is up here, that's fine too. But being dressed does kind of give you this sense of, okay, I'm here, serious, I'm ready to learn, kind of a, you know, psychology, I think. So it's just a recommendation. It's also good to carve out a space for these lectures, right? A space where you go to every time to learn. That may not be feasible, but if you can do that, then you start associating that area with learning, right? I think it's funny how when we're kids, we loathe assigned seating in our class. And then as soon as we get to be adults, what do? We go to the same desk or the same chair every day, right? Because you just associate that area as your space. So those are just kind of recommendations that might help you guys. It's really easy to just kind of, you know, go with it because you don't have to actually get up and leave your home. But the more serious you take it, I think the more easily the material will come.
I have a comment. I'm sorry.
Yes, Christine, what's up?
Yeah, so what I found last semester is that sometimes Zoom will require updates, which you don't find out till you're trying to log into the meeting. So if you open your Zoom at least 5 or 10 minutes before class starts, if there is an update, then we can get it done before the actual class start time. I got caught like twice last semester with that.
Okay.
Yeah.
So if I open my Zoom and then you guys also, or just you guys?
If you have to open it as the host, and then you can put yourself on, you know, not video, whatever, but at least it's open for us to get in. Because if there is an update that we need to get in, that's when I found it, is when it was actually trying to log me in to the meeting. So that was something I found last semester.
Great, I made a note of that. I'll try to start up my Zoom.
Awesome, thank you.
Yeah, I'll probably be on mute and not turn the camera on so that you don't have to hear me screaming.
Right.
Learn, pay attention. Yeah. So
thank you.
You're welcome. Also, it's nice, not required, but it's nice to have your cameras on. That way I can see you guys. You're not just some black box or still photo. I know that you're actually here and paying attention. I can see if you're at least, you know, pretending to laugh at my terrible jokes, that kind of a thing. It's just nice for me to see your faces. I won't require you to turn your camera on, but it's a suggestion, okay? If you don't turn your camera on because maybe your camera doesn't work or you just don't want people to see the state that you're in or your house is in or, you know, whatever, that's fine. It just might mean that I might be more likely to call on you just to make sure that you're still here with some questions. So keep that in mind. All right. Oops, why did that do that? Syllabus we already went over. Now is my opportunity to quickly plug Science Club, okay? I need recruits for Science Club. And I'm the advisor, but it is a student-run club. And our very own Emily and Elke have been former members and might still want to be, hopefully, current members of Science Club. So you can chat with them if you want to. But really, all it is you guys get together. And in the past, we've done fundraisers, and we've gone to elementary schools to do science demos like extracting DNA from strawberries. There's been field trips to the San Francisco Zoo and the Tesla factory. Of course, we can't do any of these things right now, unfortunately. So it would, until COVID lightens up, it would be more like having Zoom meetings and maybe watching sciencey movies or just having conversations about science in general. And then we're also, as a science club, we are hosting this 10-10-2020 event that will actually take place October 6th, I believe, from noon to 1. And it's focusing on climate change. And it's open to the school. I think it's open to the whole community here in Tahoe. And, you know, it's really poignant right now as we're dealing with all these wildfires and the hurricane and political denialism of science. And so if you guys want to join Science Club, shoot me a little comment in the chat, and then I'll get your name down. I need at least 10 people to make the Science Club to keep it in existence. So those of you who are interested, it's not really much of a commitment. You can show up to the meetings or not. But if that interests you in any way, please let me know. Please put your name in the chat, or I guess you don't need to put your name. Just say, I'm interested in Science Club, okay? All right. Now, this brings us to next Monday. Our lab time is going to be dedicated to Journal Club. And life is like full of important choices, and I am struggling on trying to narrow down a paper for you guys. There's just so many great ones in my opinion. So what I was going to do is I have about 4 or 5 papers here, and I just want to give you the synopsis. And if you have a paper that speaks to you, let me know. And we'll see if we can get a consensus from you guys. Again, in the chat, say, that's the paper I would like to do. And if there's one that really screams out, I may or may not pick it, but it will help me in my process here. So these aren't in any order of preference, but this paper I think is cool. It's the mucosal infection rewires TNF-alpha signaling dynamics to susceptibility to recurrence, to skew susceptibility. What that means is that this paper is in the context of UTIs, or urinary tract infections. And so you may or may not know, if you get one UTI, you're much more likely to get recurrent UTIs, especially in elderly patients. And so what this paper has done is it's found a mechanism via the tumor necrosis factor alpha that is driving that susceptibility to recurrent UTIs. Okay, and so that paper is about that and describes that. Why does it do that? So the second paper, new antibiotics selectively kills gram-negative pathogens. Okay, so as we all probably know, antibiotic resistance is a huge threat to the world, right? We have climate change, we have lack of fresh water someday that we might have to deal with, and we have pandemics, and we have this upcoming threat of all of our antibiotics being completely useless to bacteria that have evolved resistance to them. And so there's huge efforts to find new antibiotics. And this paper describes one that selectively kills gram-negative pathogens. So that gram-negative could be Klebsiella, Pseudomonas, H. pylori, and so on and so forth. And what's cool is that they actually found this antibiotic from bacteria in a little tiny parasitic nematode's gut. So it's a soil nematode. And they figured, well, if it lives in the soil, it must be encountering all kinds of bacteria, some good, some bad. Let's take some of the bacteria that resides in their gut as a resident, their microbiome, and see what that bacteria does to fight off all the other bacteria. And they found this compound in there. So that's what that paper's about. This one is the microomics, or metaomics. So omics can be genomics, looking at the genome, or proteomics, looking at protein expression, metabolomics, and meta is large, right? So it's a large analysis of elite athletes looking at their microbiome. And what they found was there's this particular bacterial species that is more prevalent in elite athletes compared to us mere mortals, and that bacterial species is associated with lactate metabolism. So if you're exercising, right, and you get into an anaerobic state, you start producing lactic acid, right, and that's what makes your muscles sore. They show that this microbe is actually metabolizing that, and so maybe that's what's allowing these elite athletes to then push that extra mile and go further. All right, so that's an interesting one. And then here's a couple other microbiome papers. They're kind of in the same vein, but these papers are looking at so for instance, the first one, the predictive power of microbiome exceeds that of genome-wide association studies in the discrimination of complex human diseases. So what that means is that rather than doing a genome-wide association where they sequence your genome and they say, okay, based on this mutation and that mutation, we think you are more susceptible to disease A, B, and C. What they're doing is they're looking at the microbiome of people and using the species diversity and the species abundance to predict whether or not that person is going to be more susceptible to disease down the road. And the microbiome has already been linked to all kinds of things like dementia and depression and post-traumatic stress. And so this is a paper that's looking at the microbiome to see how you're going to do later on. And the one down here, taxonomic signatures of long-term mortality risk in the human gut microbiome. So again, looking at your microbiome to determine, are you going to die in 15 years? Are you more likely to, or are you going to have better longevity? So those are the papers I have. Those are the ones I'm struggling on. So again, give me some chats, see if any of those speak to you. Again, so just to review, there's the recurring UTI paper. There is the new antibiotic found in the bacteria of nematode gut paper. There's the elite athlete microbiome paper, and then there's these microbiome papers associated with longevity. I'd only pick one of these two, so you wouldn't have to read both of them if you like this category in particular. Okay? Great. Okay, I know those are kind of fast, but once I figure that out, I will post the paper in week one, in the week one button. So let's see if that's, there we go. If you guys go to, is it gonna make me log in, I wonder? Oh, well, never mind. If you guys go to the homepage of microbiology, I created a blue little button that says week one. So click on that. That has the slide deck. That will have the paper that I decide on. There's some guidelines in a journal club folder that I'll show you. So you'll read this paper between now and Monday. You'll write a half page to page summary of it. Telling us what the paper's about, but then also I want you to critique it a little bit. And I don't expect you guys to be really great at critiquing if you haven't yet. For those of you who I've had in Chem 103, I have higher expectations for you guys, but tell me a little bit about the paper and your thoughts. Did you think that the paper made sense? Was it designed well? Were the experiments over-interpreted, under-interpreted? Were there good controls? Do you agree with their conclusions? And what would you do next, right? So like, for instance, with this, if we pick this paper and it was the microbiome that shows us potential for disease, what would be the next step? Well, maybe it would be looking at the microbiome of people who did not have these signatures of disease and seeing if we could somehow, in mice maybe, give them that microbiome and compare it to another mouse that has the microbiome that's associated with disease and see if that manifests in a real animal model, just as an example. Make sense? Okay. So have your eye out for that paper. It'll come up sometime today, and then you have between now and Monday to do it. But it has to be done by Monday. Yeah.
Could you email us when you get the paper that you decide on?
Yeah.
Okay.
I'll email you guys, give you a heads up that it's out there. It has to be done. Your critique has to be done before lab, right? Because that's when we're going to discuss it. So everything has to be done and turned in. Your summary has to be submitted online before lab starts so that we can all have a chat about it. Okay. This Friday's lab is going to be the growth and morphology lab. I've posted it online. And so it's there if you want to review that. The lab kit that you'll need should be ready for pickup this afternoon, okay? Or anytime between now and Friday. Now, if you are not in the South Tahoe area, I need you to remind me, because I didn't take notes of who's here and who isn't. I need you to shoot me a comment in the chat that you aren't here to pick up a kit. I know some of you have emailed me and told me, but I need to know everyone who's not able. Oh, okay, hold on. Just one second. Boys, meeting. Okay. I need you to let me know who is coming to pick up the kit and who cannot pick up the kit. Because if you can't pick up the kit, I won't put your name on the list of people who can show up to pick up the kit, all right? So just let me know. I'll have your name on a list, so you just show up to there's only one entrance where you can go by the library. They will take your temperature. You should have a mask on. You give them your name. They may ask you if you have any symptoms. If you have symptoms, fever, cough, that kind of thing, don't pick up the kit, okay? But if you're fine, then you'll just grab the kit and go, all right? For those of you who cannot pick up these kits because you're not in the area, I have also put a link to a recipe to make your own plates. So instead of agar plates, you can use gelatin and sugar and some beef stock. And instead of putting it in a plate, you can just pour it in a little dish, okay? And that's sufficient. Some people have asked if they can order plates on Amazon, which you can do. If you want to do that, that is fine with me. They may not get here by Friday, but if you can get them in by Saturday or Sunday, then that's okay too. We do need to have these done sooner than later so that we can analyze them next Friday. So next Friday's lab is you filling out the results. It should take about a week to get good bacterial growth on a plate that's at room temperature. If we put these in an incubator at body temperature 37, in a couple of days you'd have good growth. But since it's at room temperature, next week you'll analyze your plates and fill out the report and do the results. So so this lab is worth 20 points. The 10 points for this week, the 10 points for next week. Okay. And if you're having issues with getting the kit or getting the materials, since you can't get the kit, email me. We can figure something out. Okay, I know this is kind of sudden. This is the first week. And so, you know, if that's somehow not feasible to you, just let me know. Okay. Okay. That's it. Wow. We're ready to actually get into some material. Any questions before I dive in? Okay. So these are some questions I want you guys to be kind of thinking about as we go through the chapter. Because this is the stuff we're going to cover, right? What does it mean to say microbes are ubiquitous? We have to know what ubiquitous is to know that. My son knows what ubiquitous is because I use that word, and I'm feared that he also knows deleterious because that's a common sciencey word. And so I'm afraid I'm driving him towards nerdism, but maybe you don't know. Ubiquitous means everywhere. Okay, so like omnipresent. And microbes truly are ubiquitous. They're everywhere. Okay, they're all over your skin. Your hands, even if you've washed them, are teeming with microbes. Some good, some bad. Some that, if they were able to get in, could cause severe disease. Luckily, you have skin, which is a great first barrier for preventing pathogens from getting in. But you know, your phones, which you're going to swab and do in the growth and morphology lab this Friday, full of microbes. They're everywhere, okay? And most of them aren't bad. Most of them aren't good necessarily. Most of them are just doing their own thing. But hopefully, you know, by the end of this, you'll realize that that, you know, things aren't nearly as clean as we think they are. Especially, oh, okay, so here's a question. What do you think is the dirtiest thing in your home? Dirtiest meaning like the most germs.
My toilet.
My doorknobs.
Good guess. Yeah.
Sponge.
What?
Washer machine.
Washing machine. Okay.
Under my fridge.
Under your fridge? That's very dusty under my fridge, but I haven't heard it yet.
My phone.
Phone? No.
My
No?
toothbrush.
Toothbrush? Hopefully not. But that toothbrush, you know, keyboards are pretty bad. Actually, there was a study that showed that a keyboard, not like a, you know, electric keyboard, but like the keypad here in a public setting has more bacterial diversity than your toilet. Okay. Now a toilet has germs, but it doesn't have a whole lot of different germs because it gets the same repertoire. But actually, and maybe somebody said it, but I didn't hear you. The dirtiest thing in your house is your kitchen sink. If you think about where everything goes, right? It's full of nutrients, right? You wash your food in that sink, and the food that you wash off is food for the germs. You open up your chicken, your raw chicken, and you dump out that gross juice into your sink. Do you Lysol it every time you do that? Maybe some people do, but I don't. Most people don't. The sink is the nastiest thing in your home. And nasty by meaning it has things that can make you sick. So anyway, that's they did that. They determined that by doing swabs. What you guys are going to do in this lab? They swabbed a bathroom, they swabbed the toilet, they swabbed the toothbrush. And by the way, you shouldn't keep your toothbrush really close to your toilet, or you're going to be getting E. coli and all that stuff on your toothbrush. But they found that the most prevalent, diverse array of microbes existed in the sink. Okay, speaking of diverse, what is meant by diversity? Well, diversity just means the different microbial species that are present. And we'll talk about the different types. There's not just bacteria, there's viruses, right? There's fungi. There are nematodes, but worms that are microscopic. Okay, algae. Algae is not a plant. Algae is a photosynthetic microbe. Fungi is not a plant, actually. We think of mushrooms and stuff, they kind of look like plants, but they're not. So we'll talk about that. What are some of the most important events and discoveries in microbiology? So just like the history of microbiology, how did it come about? And how is the scientific method used? You guys learn the scientific method in all your science classes, and you've learned it before, but it's really important because we just kind of gloss over the 5 steps or the 7 steps, depending on what version you learn. But it's not really demonstrated how the scientific method is used in practice. And that's why I'm hoping that these journal clubs that we do and this research proposal that you will do will help reinforce just what the scientific method is. It's not just a method, it's a way, right? It's a way of discovery, of learning. So we'll get into that a little bit in the context of a microbiology experiment that was done with vaccination. And then what is the classification for microbes, and what are potential sources for new infections? All right, so those are the questions that are hopefully going to be ruminating in your mind as we talk about micro. In this chapter 1, it talks about the different fields of microbiology. So it's not just a microbiologist who's a scientist. There's all different types of applications that you can do with micro. The power of microbiology, so harvesting or using microbes to harvest different things that benefit us, whether it's using algae to produce alternative fuels, or using bacterial plasmids to produce things that we need, like insulin, for instance, or just, using yeast to make beer and bread and kombucha and all that stuff. It's very, very much ingrained into our way of life, microbes. And then a little bit of evolution. So how things went from no life to the beginning of life, and the beginning of life was microbes, right? And then they evolved over time into us, basically, right? It talks a little bit about the different types of microbes, and like I said, most of them aren't bad, but we'll talk about the good, the bad, and the neutral. And then the history. So the golden age of microbiology is considered to be 1850 to 1950. That's when Pasteur and Fleming and these guys did these foundational, seminal experiments to actually show that disease comes not from a demon or from bad air, but from actual little tiny organisms that we can't see. And then into the 1950s, when we started discovering viruses and started coming out with some vaccines that were like for polio. And now we're in kind of a second golden age right now with all of our deep sequencing methods to discover all kinds of bacterial and viral species that were not known before, because the only way to know if a microbe exists is to be able to culture it, right? So if you have a plate and it has your nutrients on there, but those nutrients aren't what are needed for certain bacteria or viruses, or well, viruses is not nutrients, it's cells, right? They need a host. Then you'll never know that they're there. But now with the advent of deep sequencing genomic material at a relatively low cost and having the computational power to then compute that to look at genomes, we can find all kinds of diversity that we didn't even know existed before. Okay. All right, microbiology. Let's start from the very, very beginning. It is the study of organisms too small to be seen without magnification, right? Now, fungi and worms, those are kind of a little bit of a stretch because some of them do barely exceed visibility with the naked eye, but for most part, it's the study of things we cannot see. We need a microscope. And then so the microorganisms that we'll discuss this quarter are bacteria, viruses, fungi, protozoa. So think of things like malaria. Malaria is caused by a protozoan. And Toxoplasma gondii, if you know about that, particularly pregnant women shouldn't be cleaning the kitty litter box if they're pregnant, because Toxoplasma gondii is a parasite, a protozoan parasite that is present in cat feces and then can be transmitted by the cat feces to the human who's cleaning out the litter box. And if you're pregnant, you are less, your immune system has been compromised because you have your own little parasite growing in you. And so you as the mother and the baby as the fetus are much more susceptible to the ravages of this particular parasite. Helminths, that's just a fancy word for worms, and then algae. So these are the things we'll be talking about this quarter. I won't spend a whole lot of time on this. This is just the different types of fields in microbiology. And if you're interested in a career in micro, I know most of you guys are going to be in nursing. So, you know, you'll have some crossover with this, but because of that, I'll just kind of gloss over this pretty quickly. We have public health microbiology. So think of CDC, which we all are very familiar with now in this pandemic. Environmental microbiology. So these guys are looking at the ecological relationships between the habitats, typically soil, but also water, and the microbes that exist in there. And a lot of, again, going back to antibiotic resistance, a lot of microbiologists are currently trying to find new bacterial species that we don't know of to try and see if they have antibiotics that they use to protect themselves from other bacteria or fungi, which was the original antibiotic. The fungi penicillium was used because they secrete penicillin. So, you know, these guys are looking at how, you know, how the interaction, the interface between environment and the microbes that make the environment up play out with each other. Biotech and industry, right? So we use microbes to make things, whether it's beer or other like foods or vaccines, vitamins, drugs, enzymes. The very first genetic engineering that was done was to make insulin. So before, insulin was purified from, I think, pigs, and used to then treat people with diabetes. And that was not a reliable source, especially as diabetes is getting more and more rampant. And so the first thing that was done was they took a plasmid. And if you don't know what a plasmid is, you will right now. A plasmid is just a circular bacterial genome, okay? So some bacteria, their genome isn't the double helix quite thing. It's just a circle, okay? And so what you can do is you can take enzymes and you can cut where those enzymes are, you can cut that DNA. And then that piece of DNA at either end or in between the flanking of those enzymes will fall out. And then you can ligate a new piece of DNA in here. Okay. And so then you can cause this genome to then start transcribing the proteins that are, you know, the genes that are in this circular plasmid. And once it gets to this new gene that you put in, it'll do the same thing. And so that's how they make insulin and a bunch of other things, by putting genes that don't originally exist in the plasmid into that plasmid, and then they purify it out. Okay. And we do that to make other things too, drugs, enzymes, vitamins, that kind of thing. Genetic engineering and recombinant DNA technology, that again is going back into the plasmids, right? And you can think of BT cotton. It's using this this essentially this toxin that's secreted by a bacterium and putting the gene that encodes that toxin into the cotton genome, so that that cotton will then also express that toxin, and that the pests that then try to eat that cotton will ingest it, right? And so when we talk about genetically modified organisms, the genetic modification is done using bacteria. Okay? Immunology is the study of the interaction of our immune system with the microbes that it encounters. Agricultural microbiology, so looking at disease in livestock and in plants. What I used to study, I discovered and characterized these viruses in a nematode, a little tiny roundworm that's non-parasitic, it's free living. And before that, people didn't know that viruses could infect worms, these nematodes, because no one had ever found one yet. And then since then, there's been interest in finding viruses in parasitic nematodes. So one nematode in particular is called the soybean cyst nematode. And so it enters soybeans and it reproduces, and it causes this cyst, which is full of a bunch of different baby nematodes, and it is detrimental to the plant. So maybe an agricultural microbiologist would be interested in finding viruses that are specific to that nematode that will kill the nematode, but not the plant, for instance. Food microbiologists, they want to make sure that your food that you purchase is not going to be contaminated with stuff, right? Basically. So from the packaging process to the delivery, the shelf life that we kind of talked about a little bit on Monday in that Labster module, that's going to be a very big part, right? Like every year it seems like there's some kind of foodborne illness that breaks out, right? Usually E. coli from packaged salad or something. And so they would be the ones that are on the site saying, okay, where did that come from? Where was it distributed? We got to shut this down. Okay. Medical microbiologists, these are going to be the guys that most of you will interact with, right? So a patient comes in, they're sick, you give them a nasal swab or something, and you send it off to the clinic, and they will analyze that to see if the patient is positive for some particular type of strep or influenza or what have you. This, I put there, so my slide decks will have tables. I put them in there for you guys to look at if you want, but most of these tables are not for you to memorize, okay? It's just there for your own information. This gives you all the different branches of microbiology and the chapter that you can find them in. So if you guys are curious in this and want to do some independent study, then by all means, take a look at the tables and look. But this is not going to be required for you to know on a quiz or test. All right, how are we doing? Any questions so far? I know I kind of breezed through that fast, but that's stuff that is just, again, I'm not concerned with all the different types of microbiologists in terms of you knowing them, okay? That's just information, okay? So any questions on that before I proceed? We're almost out of time. Okay, cool. This is stuff you will have to know. Bacteria-like organisms have been on Earth for about 3.5 billion years. Who knows how old the Earth is, roughly? Anybody?
Around 4 billion years.
Yeah, about 4.5 billion years old. So almost the entire existence of Earth has been coexisting with microbes. The first billion years, the Earth was cooling down. It went through the bombardment period where meteors and asteroids were slamming into it, and it heated up into this big fireball of hot rock and metal. And it took a long time to cool off. And as soon as it cooled off enough, then you start really seeing, not that far after, the emergence of life. We break down all life into two different categories, because what do biologists love to do more than anything else is they like to categorize things. They like to put things in neat little boxes, okay? So if you are life, you are either prokaryotic or you're eukaryotic. If you're prokaryotic, you don't have a nucleus, okay? You don't have any other membrane-bound organelles either, okay? If you're eukaryotic, you do have a nucleus. You are like us, we humans, right? You have a nucleus, you have an endoplasmic reticulum, and you have Golgi, and you have mitochondria, right? And you have all of these things that a lot of them actually came about by these cells engulfing other microbes. So mitochondria was once believed to be its own microbe that was then internalized by another microbe and co-opted to then form some of the early prokaryotes, which is kind of cool. So all bacteria is going to be prokaryotic. It doesn't have a nucleus. Its DNA is just floating free in the cell. And so you can think about it, I say pro, no nucleus, eu, true nucleus. So prokaryotic, no, eukaryotic, true. Pro, no, eu, true. That's how I kind of keep track. Here we go. I guess we should wrap it up here soon, unfortunately. But universe, about 14 billion years old, right? And about 4.5 billion, you have Earth coming into existence. And then, like we said, a billion years later, we get the earliest prokaryotic cells, which are considered archaea, not bacteria. And then over time, over a long period of time, you start finally getting into these early eukaryotic cells, about 1.75 billion years ago. So for a long period of time, these early prokaryotic cells had reign of the Earth, right? And actually, it wasn't until they started. At one point, the atmosphere of the Earth was not primarily oxygen, okay? And over time, bacteria started utilizing other food sources, and as a byproduct, they emitted oxygen. And over long periods of time, all of that oxygen accumulated enough to become the thing that life then used for their subsistence. And if you think about it, oxygen in and of itself is a very toxic element. We may not seem that way because we breathe oxygen, right? But our bodies are highly adapted to not let that oxygen impact our body, right? The rest of our body, so our molecules. Oxidation is not a good thing. Oxidation is how things rust and corrode. So the fact that these prokaryotic cells change the atmosphere is pretty astounding. A lot of people, speaking of, again, climate science, a lot of people deny the fact that humans have a role in climate change, anthropogenic change in the climate, and a lot of the argument is, well, that's just hubris, right? That's arrogant to think that we, mortal humans, can actually change the atmosphere of this gigantic planet. But if you think about it, little tiny things that you can't see changed the atmosphere billions of years ago. So for me, that's not too hard of a pill to swallow. But anyway, I'll get off my hobby horse. 2 types, prokaryotes, eukaryotes, and then of course we have viruses, because viruses aren't technically considered alive. So they're not prokaryotic, they're not eukaryotic, they are particles composed of nucleic acid and a little protein shell, and maybe some lipids and sugars if they have an envelope on the outside, but not all viruses have envelopes. SARS coronavirus 2 does have an envelope, but so viruses are the outlier. And there's prions. Maybe you guys have heard of prions. They're these misfolded proteins that once they get into your brain, they can cause other proteins that are natively correctly folded to misfold. And then you get the encephalopathy, the mad cow disease. You get like Swiss cheese brain. Okay. So there are some microbes that are not alive, but this is the main kind of categorization on the most basic level for the microbes that we'll talk about. Fungi are eukaryotes. Okay. I think that's a good place to stop, because we're now, see, this is why, I mean, an hour is, it's not very fun, because I'm just getting like into my groove, but whatever, it is what it is. So any questions right now? For today, remember to do the Labster. And if you need to reach me, just shoot me an email. I'll check my email a lot. If you have questions that you didn't pose during the lecture today, but would like to, there will be a quiz on, not this week. We'll do our first lecture quiz next week. And I've moved that to during lecture time. So Friday, at the very beginning of lecture, you'll have like 10 minutes or so to do a quiz on Canvas. So for those of you who are still having issues accessing Canvas, let me know, or navigating Canvas, let me know. I want this to be as seamless as possible. I see a lot of comments on kits. But yeah, so that's all I've got for today. I would like to keep going, but you know, it's your time, so I won't keep you. I won't get off just yet, but if you guys want to leave now, you're more than welcome to. If you want to stay on and ask some questions, by all means, we can shoot for questions. It looks like I'm just looking at papers. I see elite, athlete micro, new antibiotic, predictive power of microbiome, elite. Microbiome, elite, microbiome, athletes, elite athletes, new antibiotics. So it looks like, you guys are all really leaning toward microbiome, and that's what I expected, because that's kind of the most relevantly interesting, I think, in this day and age. We talk a lot about the microbiome. So I will send an email when I do decide on the paper. And Emily says, if you want to shoot an email, oh yeah, for Science Club. So if you do want to be part of the Science Club, we'll put you on the email list that we have going from last quarter. And it looks like I'm interested. It looks like we definitely have, hopefully maybe 10 of you. And if not, I may have to just Zoom bomb some other science classes and recruit that way. But yeah, okay, great. Any other questions?
Yes, Carl. When will the kits be ready today to pick up?
I don't know. This afternoon is what I've been told. So Kevin is the lab specialist, and he told me that they should be ready this afternoon. But I will send you an email also to let you know when they are ready for pickup. Okay. As soon as he's going to email me, and then I'll email you.
I have a question.
Yep.
When we're doing our labs, like I'm going to go do my lab next, if I'm in the middle of it and I have a question, should I just shoot you an email?
Yeah, that's
okay.
That's fine. I'll check my email incessantly, and we'll make sure. And if you guys do have any hiccups with the Labster, because they can be finicky, let me know. If you need to get a Chromebook because your device just doesn't have the power to crunch through, then let me know and I can see about getting some Chromebooks as well. Okay. But yeah, just keep in communication with me. All right. If you're having problems and I don't know, then there's nothing I can do. But because this is the first week, we'll ease into it. And if you can cruise through those labsters, great. If you're having issues, let me know. And the nice thing about them is they're not really time sensitive. You can do them later on. Are you going to make an assignment for our journal critique on Canvas? Yes, I will. I will have a journal club assignment for you to submit your critique. Okay, and again, it's just a half page, 1 page. There's also, let me, well, for those of you who want to hang out for a little bit, let me see if I can get this to work. Why? That's pages. It's probably logged me out, but there will be a good. Here we are. All right. So let me share my screen again. Where is that? Here. Okay. So, oh no, what did I do? Where did it go? Here it is. All right. So if we go to home, first of all, here's a little button down here. You click on week 1, slide deck, the labs, alternative options if you can't get the kit. I'll put the journal club paper here as well. And if you go to view all pages, not all of these are published. Let me do student view, see what you guys see. Okay. Pages, view all pages, journal club. Here are the guidelines, okay? So I choose a paper, a peer-reviewed paper. That's what's going to be on Monday. Some of the other clubs that we have, they won't be these technical papers. They'll be more like more easy reading papers that we don't really analyze. There's no experiments, but we just kind of talk about it and what are our opinions and what do we think? What are our thoughts about it? So for those, your summary will be a little bit different. For the paper this Monday, though, because it's a peer-reviewed research paper, I'll want you to provide me a little summary that gives me the salient points of that paper, and then, things like the rationale. Why is this paper important? Why did they do what they did? Who cares? Why should I care? Why should you care? Because you're going to have to provide a rationale for your research proposal when you do it. Why should I care that you want to do this? Why should the committee fund your paper? Or fund your proposal, right? So a rationale, what were some of the experiments they did? What were their conclusions? And do you agree with their interpretations? Maybe you think that they interpreted it incorrectly, or maybe they took too much liberty. Maybe you weren't convinced with the conclusions that they came to. Say that. Maybe there were some other things that you would have liked to have seen done to make the study more robust, or to answer that better. Or maybe they're, like I said before, they're lacking controls, or their data, the way they present their data, the graphical representation is wonky or a little misleading. And then what would you do next? So that's the journal club guidelines. Then if you click on assignments, there will be a journal club category for you to submit it. And I'll make it due, of course, by Monday morning, so that so that you'll all be done and have it read and we can discuss it during lab. Okay. Let's see some of the ones.
I think I'm a little
Internet access.
hazy on the lab. Is our lab lecture time in Labster? Is that what's going on? Or do we actually have like a lecture for lab as well?
So no lecture before lab. The only lectures that we'll do are during the lecture time, unless I get way behind And I feel like we really need to throw in a couple lectures during lab. But generally speaking, I'm hoping to have it to where we have our lectures and then we have our labs. And then the Monday lab is when we have our discussions together. The Wednesday lab and the Friday lab, you will be doing that independently. So Wednesdays is usually Labster, and then Fridays is going to be, this Friday is the kit. Some other Fridays will just be time dedicated to work on your proposal. Other Fridays will be more online type of labs, but yeah. Okay. Will those be due right before Monday lab? I will probably I'll make them due before Monday lecture because I don't want you to work on them during lecture, and lecture's right after lab. So it'll probably be due by 10 o'clock a.m. on Monday. And I recommend that you do it before that, but yes. And then boosting Wi-Fi, it looks like Emily said you can park on campus and use their Wi-Fi if you have issues with Wi-Fi at your home. And keep me posted about that. So if you're having issues with your connection to Zoom or to Labster or whatnot.
Yeah, so that was the deal, I believe, in the spring quarter, and I think over summer too.
Mm-hmm.
I would probably check with the school first for just like showing up, but they were letting us park in our cars in the parking lot if you're having Wi-Fi issues last 2 quarters. So I believe that's happening again, but I'm not entirely sure.
I'm almost done.
But it's worth looking into.
Okay. Well, maybe we can double check on that and see. I'll make a note.
Yeah, there is, I can't remember his name. He's in charge of the distance learning. And that's the guy to get a hold of to talk to about like getting, connections to internet for students. But it's on the LTC website under the distance learning.
Okay. Thank you, Christine. Okay. Anything else, guys?
And are we just walking into the library to grab the kits or what?
Yeah, not necessarily the library, but where the library, there's a main entrance to the library. If you guys haven't been on campus since the closure, that there'll be signs that direct you in that area too. But you walk in the main doors and there'll be people at a table and they will want to take your temperature. Just give them your name and say, I'm here to pick up a kit for BIO210. And I'm going to give Kevin the list of names. Yeah, come in. And then you just get your kit and go.
I know how to make them.
I know, but I don't want you to use fire until I can watch you guys.
Yeah, fire or gas?
Well, gas makes the fire. Okay, so any other questions?
I make them on my own all the times at home.
Okay, I'm almost done. We can do. 8-year-olds should not be using the stove unattended.
I just
What
wanted
could
to
go wrong? What's that?
I just wanted to double check if you have the list of people who can't get the kits.
I do not. I mean, I have the chat. So if you told me in the chat that you cannot get the kit, I'll look at this chat and I'll have your name that way.
All right.
Like that's the easiest way. So if you said so, then that's great.
I'm able to pick it up tomorrow. I just want to double check is any time is okay.
Yeah. You can pick it up as late as Friday.
Okay.
Because the lab is Friday. I just wanted to have it there in advance, so so that you weren't time crunched and felt like you had to rush out and get it. After lecture on Friday, if you want to go get it, that's fine. Really, all it is it's these plates, and you're going to label the plate. The plates being these petri dishes, plastic petri dishes with agar. The agar is like a jello kind of layer that the microorganisms grow on. You're going to have several plates, and you're going to just do little experiments where you're going to touch it with an unwashed hand on one side, and wash your hand and touch it on the other side with your washed hand and see what differences are a week later. Okay, you're going to cough on a plate with no mask, and then you're going to cough on another plate with a mask. And I'm assuming everyone has masks, right? And then finally, if you want, you can take a swab of your phone and then clean it. There should be I'm hoping that he has an alcohol wipe in there. Then you put a little alcohol wipe on your phone, do a second swab with another swab, with a different swab. And we're just seeing how different bacterial abundance and diversity is between cleaning and uncleaning things. Yes.
I have a question. So if I picked up the lab today, could I do that today, or do you want us to wait till Friday, because then the following Friday we're gonna like meet up on Zoom and go over it or something? Or
Yeah, well, so the way it works is that you look at the protocol if you want, but essentially whenever you start it, a week later, you're going to take pictures. You're going to take pictures after you do the things initially on Friday or today or Thursday, whenever. And then a week later, you're going to take pictures and you're going to put those in the Word document of your lab protocol. So if you want to do it today, that's fine with me. Just make sure on Wednesday, check it out. And maybe, doing it a little bit earlier might be better, because you might have better growth by Friday if you start it a little bit over a week. Does that make sense? Yeah. So feel free to start it anytime between now and Friday. And, if you want to adapt it a little bit, I'm fine with that. Like if you want to say, if you have a sponge in your sink, remember how dirty sinks are, and you want to touch half of the plate with a sponge that came out of your sink and then microwave it, and then touch the other half and see if the microwave kills off any germs or if it looks the same. I'm happy with you guys to get creative with this. It's just to give you some guidelines. But if you want to say, hey, is my sink, does it really look worse, at least on a plate, than my toilet? Then that's fine with me. If you guys want to mix it up a little bit, that's fine. Okay. And that's why it's important to, if you can't get the kits, that you are able to get something to make these plates in a timely fashion if possible. That way we can. It'd be nice to, I would like to discuss it on next Friday briefly, to see what does it look like. Does someone's hand look different from others? Because you're going to look at not only just how much stuff is on the plate, but what's the color. Different bacterial colonies have different colors. Some are white, some are yellowish, some are red. And so it'd be neat to kind of compare those from one person to another.
I also have another question about it too.
Yeah.
Where should we be storing these? Are we just gonna little petri dishes with a little lid, and I can just stack them and put them on my bookshelf, or should I leave them in a dark space or a light space? What do you recommend?
Under your pillow. No.
Perfect. That's
no, just kidding. You should have a little bit of parafilm. So it's if you're not familiar with parafilm, it's a little waxy stuff that you can stretch out. If you can't get a kit, then you can just use tape. But really all you're doing is you're going to seal the plate so they don't open up on you, and then turn them upside down and just put them somewhere out of the way, all right, where your roommate or your kids or your dog can't get to them. They don't have to be.
Will we have better results if they're in the light versus the dark, or does it just depend on the bacteria?
No, it shouldn't. It should just be temperature. So typically when we grow bacteria, we put them in an incubator and it's completely dark. So all the bacteria we deal with, and they live in your body, right? So they don't need light. But the warmer you keep them, the faster they'll grow. All right. So if you had some way to keep them warmer than room temperature, then you'll get colonies more quickly. You might get more. But this is designed to give you a week at room temperature to get sufficient stuff to be able to see.
If we put them under a lamp, will the light, like you said, it won't harm them, right? As long as it keeps it warm.
Yeah, it shouldn't. It should not. Whoops! It shouldn't harm them. No. If you wanted to give that, you don't. Ray says don't microwave them. No, that would make a mess all around. Yeah, a light like a little heat lamp should be fine. You know, your armpit's pretty warm if you want to walk around the house with the plate. No, again, joking. Don't worry, don't do that. But you know, yeah, you guys are welcome to get creative with this. Okay, I encourage creativity. It's not mandatory, but if you guys want to get a little bit experimental with this, by all means, do so. Just document it. Okay. You know, in your protocol, say I put this under a heat lamp, and if you have a thermometer, that'd be great. You could say, well, under the heat lamp, it was a little bit warmer than room temperature, so I saw colonies by this day, you know, that kind of thing. If you want to just do the street, if you want to just inoculate the plates and put them up somewhere safe and, you know, be done with it till next Friday, that's fine too. Great questions, guys.
Are we gonna use are we gonna do like enough of these projects that it would be smart to invest in plates and actually buy them, or are we?
No. I think I caught the gist of that, Isaac.
Time thing.
Yeah, if you want to buy the plates, you can, but it's really just going to be for this lab. There's one other lab that will have kits, and that will be doing grape fermentation, and all that stuff can be acquired with your local grocery store. So I'm trying to make it such that you don't have to have any expenses, and if you do, that you can get it relatively easily. Unfortunately, like if this was a face-to-face lab, yes, you would learn how to pour the plates, you would make your own plates, and you do that very frequently. But these are different times, and so we're not going to be doing a lot of that. You know, bummer, but is what it is, right? So don't feel like you have to invest in a bunch of plates. It's just for this lab. But if you would like to buy some plates or make some plates and do some experiments of your own, I may take that into consideration for extra credit, right? You know, if you feel like you don't test well, or if you feel like you've just really, your proposal is not doing well, or you're missing the deadlines and you're getting pinged for not turning stuff in. If you could design a really, you know, cool experiment looking at the microbial diversity in your home, that would be something that I would consider for extra credit. All right, anything else?
Building on top of that, I actually work at a school as a school nurse. So if you ever would like to see anything in here, like to swab anything, I'd be more than happy to do that. There's no kids right now, but you know, everything is supposed to be clean.
Yeah.
So, you know.
Yeah, by all means, if you want to start doing some swabs of different things in the school. And I mean, you know, in general, it might freak people out, but you know, honestly, the germs from one facility to the next to a home are roughly the same. They might have, you know, we all have our different microbial communities from one house to the next, and you can actually look at signatures. Like, you know, you can, if you took a metagenomic analysis of the microbiome in your home, it should look very similar to the other people that share that home with you. and as well as the bugs that are in that home. And there are slight differences between your home and my home, and the people that live in my home. And you know, I have a dog, I have a golden retriever, and he has some of the same bugs that I have. But generally speaking, you know, when you look at these plates, you might see a bunch of stuff on there like, oh, it's terrible. Like, this school needs to be shut down. It's diseased. But really, I mean, microbes are ubiquitous, right? So they're everywhere. So you give them nutrients on a plate and they'll grow. But yeah, if you have some ideas, like actually, there might be one other lab where we'll need plates, and we will have little discs, little paper discs that we will treat with certain antibiotics. I mentioned this on Monday at some point. Treat it, you know, you'll soak a disc in, say, bleach, and then soak another disc in alcohol, and so on and so forth. And then we'll take, I believe the protocol is you take a plate and you streak it with yogurt, because yogurt has lactobacillus and some other bugs on it. And so you use that to form a nice smooth lawn of bacteria on the plate. And then you put the discs down and you see which ones are the best at inhibiting it. So yes, there might be another lab that has plates in our life, but that should be it. Okay. Good questions. Thank you, guys. Anything else? Okay. Hopefully this will be fun. You know, that's the whole point. If it's stressful, then I don't feel like you learn as much. That's my philosophy anyway. Some professors have other philosophies that you need to be stressed to learn, but I like to think that we can learn and laugh at the same time. And so if you have any issues with Friday's lab, getting the materials and whatnot, don't stress out. Let me know, and we'll work it out. Okay. All right. Awesome.
Thank
Well,
you.
yeah. Sorry.
Bye.
Oh.
Thank you, Carl.
Okay, good. I'm gonna go let my 8-year-olds go play with the stove now, under supervision, and shoot me an email if you have any questions about the Labster or anything else, and I'll be on the lookout for the email notifying you about the journal club and the kits when they're ready. All right. So long.